Okay, for this next lecture, we are going to start talking about protein structures. And so we've already talked about what proteins are, and here we're going to sort of show how structure and function go together. Some of the diseases I will be covering are sickle cell anemia and cystic fibrosis, as well as just talking a little bit about our prion proteins. Now, one thing to keep in mind is that Kendrew in 1958 said that what we study is an abstraction to make comprehension of a complex entity more straightforward. So what we think about when we're learning about proteins and structures, we think of these as static. So as you learn, you think of proteins as static. And so this sort of is an easy way to explain what proteins are, but you always have to think in the back of your head that, in fact, proteins are dynamic. And cells are dynamic, and your body is dynamic, tissues are dynamic. What one cell is doing in the liver does not mean that the cell next to it is doing that same thing in the liver. So everything is very dynamic, very fluid. Also, the proteins being dynamic is important for their function because function of proteins requires flexibility within their structures and within their production, etc. So now there are different levels of protein structures. So we have the amino acid sequence shown here, which is a linear sequence. And this is primary. So primary is linear. And this is, of course, the sequence. And then secondary are going to be recurring structures. So over and over again, you'll have the same structure. And so these secondary structures are typically going to be, one of them shown here is the alpha helix. So we have an alpha helix, and the other is going to be a beta sheet. And so your proteins will fold based on the amino acid structure, the primary, into these secondary structures. And then our tertiary structures are going to fold these elements, these secondary alpha helixes and beta sheets, into 3D conformations. And typically these will have some sort of core. And the core in humans, because we're water, is usually hydrophobic. So when you go back to your amino acid sequence, where in your amino acid sequence you have your more hydrophobic, non-charged type of amino acids, those will more likely become part of that core. And then we have our quaternary structure, which is going to become a combination of 2 or more subunits, such as your hemoglobin. Okay, so why does it matter? Why do we study protein structure? Of course, many of you can probably answer this. It's all about function, but not only function. The structure really is important for our survival. Proteins need to fold properly. Enzymes need to have their active sites properly available to do a chemical reaction. A receptor has to have their proteins properly folded so that they can bind to their ligand. So there is a lot of importance going into the folding of proteins. So this is an example of cystic fibrosis. And cystic fibrosis is a lung disease, and it is multifactorial, but it's all associated with one gene. Multifactorial means that some people have some normal cystic fibrosis gene being made. It's the cystic fibrosis transmembrane conductance regulator, or CFTR. So C means cystic fibrosis, F is fibrosis, of course, and this is transmembrane. conductance, and the R stands for receptor. Okay, and this basically is a chloride channel. So again, chloride channel, you're moving charges around, and everything in our body, everything has to, especially with water movement, has to do with charges, electrolytes, where they are, where they're not, etc. So it's going to feed back into that whole osmolality. How is water moving? And so that's an important part of the symptoms associated with cystic fibrosis. Now, cystic fibrosis, of course, this conductance, the CFTR, is a gene. So you have your DNA and you have your CFTR gene. And this, of course, will become a messenger RNA. And then this, of course, will become an amino acid primary sequence. And then this will fold into its secondary through quaternary. And then you'll have some sort of function. And the function is as a chloride transmitter or a channel. Now here we're showing different patients. So we have patients A through E. And in this, you see you have different mutations. And so this is very specific. Well, this is one of the reasons that cystic fibrosis is so hard to treat, is there are so many mutations that cause this disease. And the mutations for patient A, for example, is at position 482 on the gene. And this is where you get guanine that converts to an adenine. And so this is going to change that amino acid at that when you go to translate into a protein from an arginine to a histidine. And then at patient B has another mutation, a different mutation at a different site. And this is going to generate a stop sequence, that TAG, which is a stop. And so in this patient, you don't get a full-length protein made. It's truncated because you have inserted a stop signal in the middle of the gene. You also have in another patient, you have an insertion of 2 nucleotides. And remember, our genes are read in threes, in pairs of 3. So you have 3, and this will give you an amino acid. And then you have 3, and this will give you another amino acid. And so if you shift this by 2, so now instead of this black, you now have this purple shift. So you shifted by 2, and this purple is going to give you a different amino acid. And the whole protein shifts, and this is referred to as a frame shift. A frame shift can also happen if you have deletion. So again, in this case, we'll do it in blue. In this case, you're starting here, so you deleted that one, and now you have a frame shift again for this blue, and this blue amino acid is going to be different. So that's known as a frame shift mutation. You can have deletion of 3 nucleotides, and that will not give you a frame shift like the other 2, but it'll give you a loss of some sort of amino acid in that position. And depending on where that is located, again, it's going to cause misfolding of the protein, and it can cause a dysfunctional protein. So there are many mutations that have been identified within this CFTR gene that have correlated to symptoms of cystic fibrosis. So cystic fibrosis leads to airways that have very thick and sticky mucus. And this leads to the result that infectious diseases are increased, as well as lung inflammation is increased. So you get an increase in infections and an increase in lung inflammation. And this is all in the lungs. And so you get damage to the lungs. Now, it doesn't always occur, that's not the only site, it's the lungs. So you also have this CFTR gene expression in other areas, especially in the pancreas. And so in the pancreatic ducts this chloride channel is important, again, in moving of electrolytes, moving of water. And so when you are not able to move chloride, similar to what happens in your lungs, your pancreas is affected. And so the result is that your ducts get filled again with this thick, sticky mucus, and this results in a lack of digestive enzymes. Because your pancreas will release many of your digestive enzymes. And you also have malnutrition and vitamin deficiencies because you're not able to absorb your vitamins. Now, another thing that's associated with cystic fibrosis is that your sweat becomes very, very salty. Our sweat is normally salty, but it becomes very salty. And so you have an excess loss of salt that is going to contribute to an imbalance of minerals. Again, electrolytes. And so this is going to cause dehydration. This is going to cause fatigue, weakness, general malaise, and also a low blood pressure. So the cystic fibrosis chloride channel is going to be expressed in the membrane of cells that are going to produce mucus, sweat, saliva, tears, and digestive enzymes. So that's why your lungs are infected. That's why your pancreas is infected. And again, when you present with disease, usually it's the area that's going to, or your symptoms are going to be exacerbated in certain areas. So if your lungs are affected, that's obviously going to affect your breathing. So that's going to present much more rapidly than your salty skin, because that's salty skin will have effects as well, but it's really breathing that's going to, you're going to go seek medical attention for that. Okay, so your cystic fibrosis chloride channel is an ATP gated ion channel. And what that should indicate to you that this is going to be regulated. Okay, and it has, it's referred to as an ATP binding cassette transporter. Now we have other ATP binding cassette transporters, but this is the only one that is an ion channel. Okay, and so this is a ligand-gated channel, and this is going to consume ATP during the cycle of opening or closing this gate. And so this is just an image that shows you a normal versus a cystic fibrosis. And then we have over here a sweat duct. And then we have in the middle an airway. And again, we're going to have normal and cystic fibrosis. And so basically in normal, you're going to have your ATP driven chloride channel bring chloride from your lumen into your cells. And sodium will follow chloride, so that will bring in sodium. Now in the normal lungs, you're going to release your chloride, and your chloride is going to be released into the lung tissue, and sodium is going to enter the cell, and water is also going to enter the cell. So water will follow sodium always. Now in the normal case, you're going to have normal mucus. What happens in a cystic fibrosis lung is you can't have chloride pumped out, so you're going to pump in excess sodium which is going to have excess water enter the cell to counter the sodium. Water will follow sodium. And by drawing out too much water from your lung, your airway, you lead to a dehydrated mucus, which gets really sticky. It gets kind of chunky and almost like cottage cheese type of appearance. So this is just some presentations of the disease. In this case, you have your mucus in the airways, and this mucus is not easily cleared from your lungs through coughing and things of that sort. You also have mucus secretion in your airways, and this is going to be very sticky. And then your ducts get filled with sticky mucus as well. Showing these ducts that are just full of mucus. Not all of them. You can see some that are not obstructed, but some are obstructed. All right, so the only way to treat cystic fibrosis would be to use gene therapy. This would replace the defective gene. Or add a normal gene. Okay, and so by replacing, you would knock out the bad gene and knock in a good gene, and or you can just add a normal gene. So the bad gene is still present, but you're giving them a normal genes to compensate for the bad gene. Now, of course, gene therapy is not widely used. It was used to treat some patients in the 90s. Some of those patients went on to get cancer because of the knock-in, led to cancer, a transformation of the cells, and so it was stopped. So one of the major things for treating cystic fibrosis is to control lung infection, so you give supportive care. Prevent obstruction in the lungs and in the intestines. By that mucus. For patients with advanced stages of the disease, you might have to do a lung transplant if you have advanced stages. And treating the symptoms, of course, are not going to cure this disease, but it's going to improve the quality of life of patients. And currently, patients with cystic fibrosis can live now up to about 30 years. Before, it was basically a childhood disease, and people typically would not get into their teenage years. Now, for the gastrointestinal treatment, you have to have a modified diet. And so because the pancreas is involved and the pancreas is not secreting the digestive enzymes, cystic fibrosis patients have a modified diet, and these are going to have vitamin supplements and pancreatic enzymes. And so supplements are going to be important, are fat soluble. So these are A, D, E, and K, as well as those pancreatic enzymes. You also have a high caloric content in these diets, typically twice of what's normal. And this is going to be very low in fat and high in protein. Okay, so proteins have those levels of structure. Again, we talked about primary, secondary, tertiary, and quaternary. So a primary structure of a protein is going to be defined by its amino acids, which form the actual protein. And this is, of course, goes back to the base pair sequence in the DNA. So we have DNA sequence, to messenger RNA to our primary structure. And of course, for your proteins, you'll have one end we'll refer to as an amino end, and then you have the amino acid subunits in between, and these are going to be what make up the primary structure. Now, the importance of the primary structure is going to be described by the sickle cell anemia. So with sickle cell anemia, you have a hemoglobin S. So we have hemoglobin S. And this is going to be different than hemoglobin A. So hemoglobin S is going to be caused by a mutation at a single base pair at position 6, that's the 6th amino acid in the beta globulin chain. So that's why they have that number 6 here. In a normal beta globulin chain, so this is our beta globulin, that position 6 is held by a glutamic acid. And our sickle cell number 6 position, that is going to be a valine. And this is because there's a single point mutation from GAG, get a different color, okay, to GTG. And by changing that second GA, by going from adenine to thymine, then you're going to change the amino acid from glutamic acid to valine. Glutamic acid, of course, is very hydrophilic, and valine is very hydrophobic. And so one of the first reports of sickle cell anemia was back in 1910 in a journal where they saw pear-shaped red blood cells. And of course, we now know these as sickle cell shaped. This sequence where you change the amino acid that leads to another amino acid is called a missense mutation, because you're really changing you're going to change ultimately the structure of the protein because of this change. And so this is what really happened. Again, in your normal DNA, you have GAG to get glutamic acid. The mutation, a single point mutation from an A to a T, and now you have valine. So you get a uracil instead of an adenine, and that changes the overall structure of the hemoglobin. Now, a really easy way to detect hemoglobin S is to run a gel electrophoresis. And gel electrophoresis is where you basically have a charge across an aqueous solution, and you immobilize proteins in a gel, and then they will move across the gel based on their charge. So for example, we'll have a negative charge here and a positive charge here across the gel. You will have your start wells. So this is going to be our start. And this is where you will load the sample using a pipette tip. So this is a pipette. And you load the sample. And then you turn on the charge. And so, of course, opposites attract. So anything that is more negative will run towards the positive, and anything that is more positive will tend to stay more or closer to the negative. And so here we have a band, a black band here, and we'll have a black band here. Okay, and then here we'll have a black band. And then here we'll also have this black band. All right, so we know here that this is normal. This is our control. So this is normal. And this will be hemoglobin A by hemoglobin A. And so you can see here that we have two different outcomes depending on the different patients. So patient 1 does have a band that coincides with normal hemoglobin A. So patient 1 will have hemoglobin A, but patient 1 also has the second band here, which is not in the normal, so this is the hemoglobin S. Patient 2 only has one band at hemoglobin S, so patient 2 would be a homozygote for hemoglobin S, whereas patient 1 is a heterozygote. Has one good hemoglobin and one S hemoglobin, and this is a homozygote. And of course, the control normal would also be a homozygote, but for hemoglobin A. Okay, so that's how you can tell on a gel very quickly if your patient has hemoglobin S. All right, now moving on to the secondary structure. The secondary structure is going to describe the way that the amino acid folds into itself due to intramolecular hydrogen interactions. And two common secondary structures are going to be the alpha helix and the beta sheet. So the alpha helix looks like a helix, what we're used to, DNA helix, etc. And the beta sheets can have many or two types of folding, which we'll talk about in just a moment. So our alpha helix is a secondary structure, and these are going to have localized interactions that have a repeating element. They're usually very uniform. They usually are spaced apart equally. These are going to be rigid, stable, and compact. Now also, there are going to be common elements of our globular proteins. So you will find alpha helixes in globular proteins. You're going to find them in membrane spanning domains. And that's going to be in the protein. And our DNA binding proteins are alpha helixes. Okay, one thing to keep in mind with our alpha helixes, I don't think I have it here, but proline. Oops, spelled that wrong. Proline is known as the alpha helix breaker. And we talked about proline a little bit in our amino acid lecture, and we talked about how proline has a unique cyclic structure that makes it very, very rigid. And so it tends to bring too much rigidity to the alpha helix, and the alpha helix cannot fold properly, and it actually breaks. Now, what proline can do, so where do we find proline? We're going to find proline in areas that join alpha helixes to beta sheets. So proline can be important. Again, it's not going to be in the alpha helix structure, but it's going to be there to link together different secondary structures, the alpha helix and the beta sheets. And also tripolines are going to be important in protein-protein signaling. And so this is going to, prolines are also going to be common in collagen. All right, so just another side note about alpha helixes is there are some amino acids that favor alpha helixes. So we have amino acids that are friendly, and these are going to be alanine, aspartic acid, glutamic acid, isoleucine, and leucine. So these are friendly. And then we have amino acids that are foes to an alpha helix, and these are going to be proline and glycine. Glycine bends too freely, and glycine will deform the alpha helix. So it's not rigid enough. So you have proline, which is too rigid, and then you have glycine, which isn't rigid enough. And both of those are going to be excluded from alpha helixes. And also keep in mind that alpha helix is a spontaneous folding of the protein. It gets stabilized by hydrogen bonds between the amide nitrogen and the carbonyl carbon of the peptide bond. So it goes back to the peptide bond and the interactions of the side chains of the amino acids that form the alpha helix. All right, so next up is our beta sheet. So there are two forms of beta sheets. There's something called the anti-parallel, and then you have the parallel. And so most are going to be anti-parallel, so most of our protein beta sheets are going to be anti-parallel. Some are going to, and then a lot are going to have mixed parallel, anti-parallel. So I'll put or mixed. And it's rare to only have parallel. And so you can see that the anti-parallel is more organized. So you can see that it follows more organization, whereas the parallel, because of the way that it is folding onto itself, it has to do a crossover. And so this is not as organized. All right, so beta sheets also are going to be stabilized by hydrogen bonds. And these, of course, are in the actual amino acids. It's the amide nitrogen with the carbonyl carbons. That's peptide bond. Now your beta sheets have a little bit more requirements. So the beta sheets are going to have to be made up of two or more different regions of a protein. And at least 5 to 10 amino acids for each region. Okay, so now we have some super secondary structures that are discussed. We have something called a helix turn helix motif of a protein, and this is where the amino acid sequence causes a turn. So this is an alpha helix, turn, alpha helix. That's where it gets its name, helix turn helix. And these regulate gene transcription in bacteria. So bacteria don't have as many enzymes as we do. And so basically what that means is that they have to have unique ways to turn on and off genes. One way to turn off a gene is to simply make it so the polymerase falls off. And the polymerase falls off, then you can't continue. So that's a way to control regulation of genes. Now, another one is going to be a helix loop helix motif. And this again, you have your alpha helix here, and this is a loop that forms, and then another alpha helix. And we also have what's called a zinc finger, in which you have a zinc incorporated, and that is referred to as a zinc finger. Helix, loop, helix, and zinc fingers are eukaryote transcription regulators. All right, so moving on into tertiary structure. So a tertiary structure is going to be a structure that is going to interact by various mechanisms. The backbone of the protein interactions are going to define most of the secondary structure interactions, and the side chains of the interactions are what are going to define the tertiary interactions. So for your protein, you have your backbone interactions, and this is your peptide bond plus hydrogens, and these are going to define your secondary structures. And then the amino acid side chains define your tertiary interactions. Now, forces that are going to control these are the forces that were already talked about. We have our hydrophobic forces, our hydrogen bonding, electrostatic, other van der Waals forces such as dipole-dipole, which is similar to hydrogen bonding, and our London dispersion forces, which are our weaker forces. And so all the amino acid side chains interacting with all of these forces are what are going to create that tertiary structure. So tertiary structures are going to have longer range interactions, and they are going to involve those amino acids within the secondary structures. And you get something called structural domains. You also get a sort of localization of your different amino acids with respect to protein folding. So your hydrophobic, are going to localize in the interior. And your hydrophilic are going to localize on the surface. Where they will interact with water. Now, some examples of these proteins are transmembrane proteins. And so transmembrane proteins are going to span a membrane. So here we have a membrane, and here we show a transmembrane protein as 1, 2, 3, 4, 5, 6, 7. So this is a hepatohelical protein. It's also a G protein receptor. And they are going to be important for transporting proteins. They're going to be hormone receptors, ion channels. They're going to always have alpha helical regions in which you have your hydrophobic amino acids exposed to the lipid bilayer. So these are our alpha helix. And the alpha helix will fold so that you have your hydrophobic interacting with the lipids. And then your hydrophilic sequences are going to be often in the aqueous environments. So, of course, this is your aqueous environment, so these will be hydrophilic. All of these will be hydrophilic, etc. And so you're also going to have post-translational modification of some of these. You can have sugars added, which are carbohydrates shown here. So this will be post-translational modification. And just as a reminder, remember DNA to messenger RNA is transcription. Messenger RNA to protein is translation. So post-translational, so post-protein production modifications. Then we move on to quaternary structure. These are going to be held by those same forces by tertiary structures, those van der Waals, dipole, etc. So quaternary, you have the same forces as tertiary. The proteins are often going to have multiple monomers, and these are referred to as oligomers. A homo-oligomer is multiple identical polypeptide chains. So we have a homo-oligomer. And an example of this is collagen. And then we have hetero-oligomers. And this is where they are different. An example of this is hemoglobin. So you have alpha and beta, so those are going to be a hetero. Whereas in collagen, you have the identical polypeptide chains over and over again, and those are going to be homo-oligomers. Now, quaternary structure has some things going on with it. You have instances of cooperativity. So remember, you have multiple peptides in a quaternary structure. And so some of this is due to making it sure that your protein really wants to function. So hemoglobin has cooperativity. Binding of that first oxygen to hemoglobin can bind 4, takes a lot of effort. After that first one is bound, then the rest of the molecule opens up and it's easier to bind the rest of the 3. You also have co-localization for this function. So quaternary structures of proteins are often going to be localized together so that they can function in some sort of reaction that they are needed. We also have combination. And so your immunoglobulin will have different responses, multiple receptor responses. You have structural assembly, your actin. Your actin is going to give shape to your cells, and it's also going to provide form and function to your cells. Okay, so that's all.